Hello everyone. Today in this video, let's discuss about the topic called Kingdom Monera. Before dive into the topic, let's discuss about the R.H. Whitaker's Five Kingdom classification. So, usually it is very difficult to study each and every organism's body, their morphological structure, their internal organization, their habitat, their nutrition, and all. So, in order to make it simplify to study, R.H. Whitaker classified all the organisms and placed the similar type of organisms in one group and another set of similar organisms in another group. For example, if you consider the Monera, Monera is a kingdom in which all organisms are having similar type of organization, like similar type of characters. For example, Monera is only having bacteria. Only the bacteria are present under the kingdom Monera. So here if you see the bacteria, all the bacteria are not having any nuclear membrane around the nucleus. So that is the characteristic feature of the bacteria. So all the organisms present in the Monera are not having any nuclear membrane around the nucleus. So uh, they are sharing a similar characteristic, right? So here similarly, protista, all the single cellular organisms are placed under the kingdom called protista. So every kingdom is having a similar type of organisms. Likewise, so that we can study easily, right? We couldn't study each and every bacteria, so collectively we can study a bacterial structure, but that will be so easy, right? So that is why R.H. Whitaker classified organisms and placed in different kingdoms. So let us first look at the kingdom called Monera. So what is Monera? What are the organisms present inside the Monera? So organisms present in the Monera are called as bacteria. We can call them as prokaryotes also. So what is prokaryotes? So, prokaryotes means, let us see the difference between the prokaryotes and eukaryotes. So, prokaryotes are the organisms which are not having nuclear membrane around the nucleus. So, the cell which is not having nuclear membrane around the nucleus are called as prokaryotes. And the organisms which are having the nuclear membrane around the nucleus are called as eukaryotes. Okay, so the bacteria are the only organisms which are not having nuclear membrane around the nucleus. So they are also called as prokaryotes. Prokaryotes or bacteria. Prokaryotes are also called as bacteria, and bacteria are also called as prokaryotes. So eukaryotes means apart from bacteria, the rest of all, that means protista, fungi, plantae, animalia. So organisms present in all these four kingdoms, protista, fungi, plantae, animalia, or comes under eukaryotes because these organisms are having nuclear membrane around the nucleus. So Monera or or comes under prokaryotes, protista, fungi, plantae, animalia comes under eukaryotes. This is the basic difference. So next, so we're done with the term called prokaryotes, and then comes habitat. So if you see the bacteria, um, where the bacteria lives. So bacteria can live in the soil or air or water or hot springs or deep oceans, deserts, etc. So they can live almost everywhere, right? So since they live in everywhere, they are called as cosmopolitan organisms. So some bacteria called as archaebacteria, they can live. They are called as archae archaebacteria. So this archaebacteria can live in extreme conditions like hot springs. So hot springs are extremely hot, right? So we couldn't survive there. But this archaebacteria can survive at that hot springs because of some modifications. They can survive their body modification, they can survive in that extreme conditions also. And also they can survive in archaebacteria can survive in extreme salt conditions and wetlands, like for example methanogens. methanogens are present at the like in the um, in the elementary canal of the like cattle, elementary canal of the buffalo. That is why methane gas is producing from the dung of the uh, buffalo. So yeah, in that conditions also they can survive in such conditions. Yes, and some bacteria can also act as parasites. So this is the habitat of the bacteria. They are called as cosmopolitan organisms because they can be present almost everywhere. Yes, then the size of Monera. Coming to the size of Monera, it is 2 to 5 micrometers. But here, the most important thing is Mycoplasma. This is the smallest bacteria of all. This size is 0.3 micrometers. And also, another important thing is it doesn't have any cell wall. Generally, the prokaryotes, all the bacteria is having a cell wall, but Mycoplasma doesn't have any cell wall. This is the most important thing you should remember. And this is the smallest bacteria too. And then comes shape. So let's have a look at shape of the bacteria. So some bacteria appear spherical in shape, and if they and some bacteria appears rod shape, some bacteria appear spiral in shape, some bacteria appears comma shape. If they present spherical in shape, they are called coccus. If they are in rod shape, they are called bacillus. If they are in spiral shape, they are called spirillum. If they are comma shape, they are called as vibrio. So, coming to the coccus, if it is singular, we call it as a coccus, but if it is present two in number, then they are called as diplococci. Diplo means two, and cocca is the plural term. Next, if they are present in four in number, they are called tetrads. If they are eight in number, they are called sursine. If they are appear like chain like appearance, then they are called streptococci. If they are many in number, then they are called as staphylococci. Similarly, bacillus. Bacillus is generally rod shape. If they are plural and they are present in chain-like manner, then they are called as streptobacilli. So this is all about the shape of bacteria. These are the terminology you should remember. And then comes the structure of the bacteria. See, coming to the structure of bacteria, here the bacteria is having a cell envelope. So here the outermost layer is called as a glycocalyx, right? This outermost layer is called as glycocalyx, and inside this layer there is a layer called as a cell wall. Inside the glycocalyx there is a layer called cell wall, and inside this cell wall there is another layer called plasma membrane. So all this plasma membrane, cell wall and glycocalyx, all these three together called as a cell envelope because it is protecting the cell. Yes, and then comes inside. If you go inside the cell, then we can see the cytoplasm, and inside the cytoplasm there is a genetic material called nucleus. And we have already said that prokaryotes doesn't have any nuclear membrane around the nucleus. Yes, so nucleus is freely present in the cytoplasm without any nuclear membrane. And along with this nucleus there are some ribosomes and also plasmid, inclusion bodies, mesosomes are present. So some external structures are also present along with this. They are called as fimbriae, sexpili, flagella. So now let us discuss about each and every organelle and cell envelope detail in detail. So yes, the first one is cell envelope. This cell envelope is composed of glycocalyx, cell wall, and the plasma membrane. So we said that all these three layers together called as cell envelope. So let us see the first layer, glycocalyx. This glycocalyx is made up of mucopolysaccharides. Okay, and if this layer called glycocalyx is present tightly, then this glycocalyx is called as capsule. If the glycocalyx is arranged loosely, then it is called slime layer. Okay, yes, this is all about the outermost layer, glycocalyx. Then comes to the cell wall. So this is the cell wall. composition is peptidoglycan. So cell wall is made up of only the bacterial cell wall. Okay, not all cell wall, all organism cell wall. Only the bacterial cell wall is made up of peptidoglycan. It is very important thing you should remember. The cell wall of bacteria contains peptidoglycans, and another important thing is mycoplasma, that is the smallest bacteria. It doesn't have any cell wall. This is another thing you should remember. And then comes the plasma membrane. So the plasma membrane is pretty much similar to the eukaryotic cell. If you see, I have drawn here the plasma membrane, and actually I've drawn a line, right? But it is not appears like a line. If it is actually appear like this, if you zoom in this line, it will look like this. So it is composed of, plasma membrane is composed of phospholipids. Phospholipid molecules. Okay, so phospholipid molecule is present like this. So all the phospholipid molecules collectively present and form a layer, and they appear in two layers, they are arranged in two layers. So this is called as a phospholipid bilayer. The plasma membrane is called as phospholipid bilayer. Okay, so if you see one phospholipid molecule, it has a hydrophilic head and the hydrophobic tail. It has head and tail. Head is called hydrophilic head and tail is called hydrophobic tail. Okay, then yes, and between the head and tail, there is a bond, is connection. There is a connection between the head and tail, and this is called as ether, sorry, ester bond. Ester bond is present between head and tail. In case of archaebacteria, you should remember, archaebacteria, between the head and tail, ether bond is present. Ether bond is present between the head and tail. In case of rest of the bacteria, like eubacteria, remaining bacteria, so actually bacteria are divided into two types: eubacteria and archaebacteria. So archaebacteria is having a ether bond between the head and tail. In case of eubacteria and also in eukaryotes and eubacteria, between the head and tail, ester bond is present. Okay, this is the difference between archaebacteria plasma membrane and the eubacteria plasma membrane. So yes, this is all about the plasma membrane, and then comes the nucleus. So yeah, nucleus is actually a circular in shape. It is actually circular DNA, and it doesn't have any nuclear membrane around the nucleus. And then comes ribosomes. Ribosomes are actually 70s type of ribosomes are present in prokaryotes, while in case of eukaryotes 80s type of ribosomes are present. And this 70s type of ribosomes are having two subunits called 50s and 30s subunit. Here one more important thing you should remember is S for Swedberg units. This is also the important question they will ask, that is called Swedberg unit. S represents Swedberg unit. So 50 years is the larger subunit, 30 years is the smaller subunit. This type of ribosomes are present inside the prokaryotes. And then comes the inclusion bodies. So inclusion bodies are also present in the bacterial cells. So these inclusion bodies are actually for the storage purpose. So these inclusion bodies, some of them store glycogen, they may store starch, they may store sulfur and the phosphorus. So if they store glycogen, they are called as glycogen granules. If they store starch, they are called starch granules, or similarly sulfur granules and the phosphorus granules. So these type of granules are present in the cytoplasm of the bacteria. And then comes plasmid. So here the plasmid is the the plasmid is actually present in only some type of bacteria. It is not present in all type of bacteria. It is actually having some genes. Plasmid is having some genes. So these plasmid, if is responsible for formation of sex pili in that particular bacteria, and also this plasmid helps in showing resistance to antibiotics. So this plasmid is nothing but extra chromosomal DNA. That means nucleus is also present, right? That is having genes, but apart from the nucleus there is also some another thing called plasmid. It is extra, called as extra chromosomal DNA, and it have genes. So these genes are responsible for have showing resistance to antibiotic, and also these genes are helpful in formation of sex pili. For example, if you see a bacteria which is having a plasmid along with the nucleus. So the bacteria which is having plasmid along with the nucleus shows resistance to antibiotics. If you give any antibiotics, this bacteria show resistance because uh, it have genes in the plasmid that shows resistance to antibiotics. And also this particular bacteria which is having plasmid shows a sex pili because these have genes that help. There are some genes present in the plasmid that shows, that helps in formation of sex pili. So if you compare this bacteria with the other bacteria which doesn't have any plasmid, only the nucleus, then that bacteria doesn't show resistance to antibiotics and doesn't show any formation of sex pili. Only the bacteria which are having plasmid can show resistance to antibiotics and having a sex pillar. They have sex pillar. So this is the difference. And the bacteria which are having a plasmid are called as donors, and the bacteria which doesn't have plasmid are called as recipients. Next, mesosomes. Mesosomes are nothing but the infoldings of plasma membrane. So if you see, this is the plasma membrane, and this plasma membrane have some infoldings. So this plasma membrane fold inwards. So these are called as infoldings. So these infoldings may be appears in three forms. One is vesicle, another one is lamella, and another one is tubular. In these three forms they may be appear. So these mesosomes are mainly useful during the cell division. So during the cell is dividing, then the genetic material comes and attaches here at the mesosome. So during the cell division, these mesosomes helps. And um, and also one more important thing is here there are some enzymes are present that helps in respiration of for this bacterial cell. It helps in respiration. And then comes fimbriae. Fimbriae is used for the attachment of the bacteria. So here if you see the bacteria, it have some structures called fimbriae, and this fimbriae is helpful for the attachment to the substratum. If bacteria wants to attach to any substratum, this fimbriae helps. Then comes sex pili. This sex pili is used for conjugation. I have already said that this plasmid, if the plasmid is present, then it helps in formation of sex pili, right? So what is this sex pili and how it will helps? See, I will draw here. If the plasmid is having, like if the bacteria is having plasmid, then it will helps in formation of sex pili. And another bacteria which is not having any plasmid, and that doesn't have any, only it have any it has nucleus and it doesn't have any sex pili because it doesn't have plasmid. So now what happened, this is called as a donor cell and this is called as a recipient cell. So now what happened? This donor cell which is having sex pili will form, with the help of the sex pili, it forms a conjugation tube with the recipient tube. So between the donor and the recipient, the donor cell forms the conjugation tube between the donor and the recipient cells with the help of sex pili. So now what happened, once the conjugation tube is formed with the help of sex pili, now this donor cell will donate. So here it also have the nucleus. Now this donor cell will donate this plasmid to this recipient cell. Now the recipient cell will get the plasmid, and now the recipient cell will become donor. Okay, so this is the transfer of genetic material. This process is called conjugation. This is one method of transfer of genetic material. So here during the conjugation, this sex pili will help. So yes, um, this is all about. So we have done. This is all about the prokaryotic cell structure. So we have done with shapes and structure, and also we have done with the RH Whitaker 5 kingdom classification, prokaryotic versus eukaryotes, and the habitat, size, shapes, structure, and all. So, yes, this is all about the prokaryotic structure. If you like this video, please do subscribe, like, and share. Thank you.